എപ്പോഴാ പറയുന്നത്?
Annama madam, you can unmute and start.
സ്റ്റാർട്ട് ചെയ്തോട്ടെ?
You can start.
Okay, madam.
സ്റ്റാർട്ട് ചെയ്യട്ടെ, പറയട്ടെ. വിജയമ്മ പറ. അല്ല, ഞാൻ പറഞ്ഞോട്ടെ, തുടങ്ങട്ടെ.
Annama madam can start.
Good afternoon.
A very good afternoon to all. Warm welcome to all those who have joined Zoom meeting. Today's topic is educating the ever-changing brain, innovative teaching, learning methods rooted in neuroplasticity. This is presented by our Dr. SARITHA SHENOY. She studied her MBBS at Trivandrum Medical College batch of 1993 and completed her PG from Kottayam Medical College. During 2004 to 2007. She showed exceptional academic performance during her PG days and still continuing that. Kottayam alumni PG classes for physiology was taken by Dr. SARITHA for a long time. In short, SARITHA acts as a pinch of salt which adds flavor in all programs at Kottayam Medical College and CME all over Kerala. She is a gifted spokesperson and mentor of MBBS students. Our last CME at Bcmch 2024 Osteophysicon, SARITHA was a speaker. Now, VIJAYAMMA, Vice Principal and Professor of Anatomy at Bcmch. She is my friend and colleague for a long years, both at Kottayam and Bcmch. She joined MBBS 1978 batch at Kottayam MCK, worked at Anatomy Department Kottayam for a long time and at various other medical colleges. And after retirement from Kottayam, she is at Bcmch Medical College. Now she is the Vice Principal. She excels both in academic and administrative roles. She is a real compassionate mentor to students. Dr. VIJAYAMMA is our moderator of the Zoom meeting. Now I kindly request VIJAYAMMA to take over.
Good afternoon, all. Can we start the topic now itself? So we can start the topic of innovative teaching and learning method rooted in brain neuroplasticity is our topic. So we can start the topic. Over to SARITHA.
Hello.
Yes.
Hello. Shall I start, madam?
Yes, yes.
Am I audible? Okay. Thank you, Annama madam and VIJAYAMMA madam, for this wonderful opportunity. It is the third time I've been invited at this prestigious institution of Believers Church Medical College for a CME talk. Today's talk, what is given to me is to discuss regarding the newer teaching learning methods and connect it to neuroplasticity. So that is connecting physiology with medical education. I don't know how far I can be provide justice to this connection. I'll try my level best. And thank you, VIJAYAMMA madam, for challenging me with this task. Okay. So we shall start first regarding the synaptogenesis. When we are born, it is said that all of us are born rich. Okay. It is not in terms of money, but it is in terms of the number of neurons which we have in our brain. So before birth itself, the neurulation and neurogenesis starts, and it is said that approximately around 86 billion neurons are there in our brain at the time of birth. Okay, but these neurons develop synapses only later in life. More synapses are formed later in life. And as after birth, the synaptogenesis more number of synaptogenesis begins. And by 2 to 3 years, it is said that there will be around 1,000 trillion connections in brain. So based on the number of inputs received to the brain, the synapses develop. Okay, so by 2 to 3 years, the amount of synapses which is there in the brain will be of maximum amount. So at the time of birth is the time when we are having the maximum number of neurons. Later on, the number of neurons reduces as a part of aging, but the synaptogenesis begins later in life, and it is maximally seen after 2 to 3 years. But what happens later is that's what is the concept of what is called as born rich. So first synapse is developed at 20 weeks of gestation, but the amount of synaptogenesis peaks by 2 to 3 years. And before birth, there will be more synapse seen in spinal cord and brainstem. That is for basic reflexes to develop. Later on, after birth, more synaptogenesis happens in our sensory system, special sensory system like visual system, occipital cortex, auditory cortex, and also the prefrontal lobe. Okay, massive overproduction occur in terms of synaptogenesis by 2 to 3 years. But later on, what we see is that there is also a reduction in the number of synapses formed. So when we are born with maximum number of neurons, and in the early part of our lifetime, there will be maximum amount of synapses formed. But later on, what happens is there is reduction in the number of neurons, and there is reduction in the number of synapses also. Now, this is a picture just showing the number of neurons when we are born at 1 month and 9 months. And you can see this number of synapses are increasing from the day of birth to maximally reaching a number by 2 to 3 years. In adulthood, as a person moves into the adulthood, the amount of synapses comes down. The number of synapses comes down. This is what is called as synaptic pruning. Okay, only, or what is called as use it or lose it. Only those synapses which are frequently used, which stays in our brain, and those synapses which are not used is lost from our brain. Now, same picture once again. At birth, there will be minimum number of synapses. As age advances, by the age of 2 to 3 years, there will be maximum number of synapses, and later on, the number of synapses also comes down. Okay, so there will be elimination of less used synapses. This is called as synaptic pruning. The early pruning is done by genes, genetic influences. Later on, it will be done by our experiences, epigenetic influences. Okay, so both genetic influences and epigenetic influences play a role in pruning of our synapses. Now, is it important? Do we need to prune our synapses? Maximum pruning occurs at 2 to 10 years of synapses. But in our prefrontal lobe, the pruning continues, and it is seen later in adolescence. So it is said that the adolescent age group are now here I would like to link learning, teaching with synaptic pruning, because we, as the first MBBS teachers, we are catering to a group of students who are just after their adolescence age. So at the age group of 19-20 is what they are coming to us. And that is the time where pruning begins and pruning is seen. But the pruning is not complete. Okay. So based on the life experiences which they receive, the pruning will be completed and their synapses will be made more refined. So we should not blame our students for showing all those inexperienced responses. Blame it on the synaptic pruning or synaptogenesis, which is still going on in their brain. Okay. So this pruning also is first seen in the visual and auditory areas, and prefrontal lobe, which is the lobe which is concerned with our mainly our social behavior. The pruning occurs during the late adolescent time. Okay, now who does the pruning? This pruning is done by the microglia, which is one of the neuroglial cell, or it is also called as the scavenger cell. It is called as the brain sculpture. Okay, so this microglia has, with the help of certain find me and eat me pathways, does a survey around the brain and finds out those synapses which need to be pruned and eats those synapses which are not required for our normal activities by that person at that time. So this find me, eat me pathways is mediated through certain chemokines and complements. So chemokines CX3CL1 is the ones which helps us to find those synapses which are not utilized now and then, and those synapses are degenerated with the help of complement system. Okay, so this process is continuously going on in our brain based on our genetic influences and based on our epigenetic influences, that is our environmental factors. Now, is it important? Yes, it is of course important. Now, exposure to the environmental and genetic factors early in life to this microglial pruning mechanisms like pollution, sensory deprivation, stress, dietary irregularities, infections prone, if the child is prone to infections, or if there is some lack of cell signalings or growth factors or this complement systems, that is genetic factors, will alter this microglial activity and makes the person vulnerable for development of developmental disorders like autism spectrum disorder, schizophrenia, etc., and also a variety of psychiatric disorders like psychosis, OCDs, anxiety, depression, dementia later in life. Okay, so this pruning, neurogenesis, synaptogenesis, and neuronal pruning, synaptic pruning, is a process of evolution, and that occurring in the correct pathway is very important for our normal mental functioning. Okay, so now based on this, what we have understood is that our brain is growing not in numbers, but the growing is occurring in terms of connections, synapses or connections, and those connections which are frequently used by us will remain in our brain, and those connections which are not frequently used by us undergo degeneration. Or in other words, brain is continuously getting, what you call it, changed. Continuous change is occurring in our brain, or it is continuously getting molded. This is what is the concept behind synaptic plasticity, okay, which was put forward by synaptic pruning. Before moving on to synaptic plasticity, we have already understood why the synaptic pruning is important. The significance of synaptic pruning is to develop a brain which is more refined and more efficient to function. And this is the basis behind learning, memory, and emotional regulation. So new things which we learn helps us to create new synapses. And those things which we are forgetting is because degeneration of those synapses which we have already created. Okay, so this also helps us to understand the concept of pruning targeted therapies, and also helps us to modify childhood behaviors. Okay, so pruning is a part of evolution, and understanding the concept of pruning will help us to understand the process behind a lot of developmental disorders and psychiatric disorders. Okay, so with this short discussion, we have understood that our brain undergoes molding, or that molding is what is called as plasticity. Okay, those synapses which function or which are frequently stimulated remain. those synapses which are not frequently stimulated gets degenerated in our brain. Now, when it comes to the synapses which are remaining in our brain, they are the basis for our memory processing. As I have said, this pruning mechanism and wiring mechanism, okay, synapses forming together mechanism is very important for learning and also memory consolidation. If you look at a blueprint of memory processing, the memory processing goes on to the various process. When we receive various stimuli, first these stimuli will be encoded in the form of neuronal signals in our brain. Okay, so that is called as encoding. Whatever stimuli we are receiving, those stimuli will be encoded in our brain in the form of neural signals. Now, this stage is very critical in memory processing. Why it is very critical? Because this is the first step of memory processing, and based on this step, the further steps of memory storage happens. Okay, it is said that a lot of motivation and concentration, if is given to this stage of encoding, the consolidation, later steps of memory processing, that is the consolidation, storage and retrieval of that fact becomes more clear and more better. If your encoding is not proper, then consolidation will be not proper, storage will not be proper, and your recall capacity or retrieval of that memory is also not proper. Okay, so for encoding, we need synaptic pruning and we need plasticity of synapses. Okay, right? Now, this property of synaptic plasticity was put forward by Donald Hebb, and it is called as Hebbian postulate. Simply means that neurons that wire together, means like if you see something, you see a water bottle, a blue water bottle in front of you. Okay, so that blue water bottle gives you an impression of blue color. Water bottle is used for drinking water. Okay, and you get the feeling of thirst, and looking at the blue water bottle, you will feel that yes, I have to drink water. That tendency of drinking water arises in your brain. Okay, so just by looking at a blue water bottle, what happens is that so many neurons placed in so many areas in our brain starts firing. Okay, so that is what happens. And whenever you look at a blue water bottle or a water bottle, these neurons which had started firing keeps on firing repeatedly, and they wire together, or a circuit is formed between these excited neurons. That is what is called by the Hebbian postulate, neurons that fire together and wire together. Along, there is coincident presynaptic and postsynaptic activity that leads to persistent change in synaptic efficacy. Okay, so this is what I was describing. Our synapses, our brain functions in circuits, and those neurons which fire together are connected with the help of circuits, and those circuits which are continuously working becomes not dominant, and those circuits which are not continuously utilized become non-dominant. Okay, this is the foundation of synaptic plasticity, and this is the foundation behind the cellular mechanism of understanding, learning, and memory storage. Okay, so what happens in our long-term memory? So whenever we see a blue water bottle, what happens is that, okay, we get the memory of this is a water bottle, this is blue in color, we can drink water from it. All those memory will be retrieved from our brain. Okay, so this is the concept of long-term memory. In long-term memory, the synaptic event which is happening is called as long-term potentiation. This is the primary process of encoding, storage, and associative learning which is happening in long-term memory. When you stimulate a synapse persistently with high frequency stimulation, okay, so certain synaptic changes occur in our synapses. Okay, this changes mainly occurs in the hippocampus, mainly in the pyramidal cells of CA3 and CA1. The net effect of these changes is that there will be increased neurotransmitter release and increased receptor sensitivity. Okay, so the synaptic event occurring in long-term processing of memory is called as long-term potentiation. This long-term potentiation occurs in two stages, early LTP and late LTP. Early LTP changes occur within seconds of stimulation and it remains for few minutes in our brain. Late LTP changes occur in hours and it sometimes takes some days for the changes to happen. Okay, so now if something goes wrong during this process, the early LTP changes will not be converted into late LTP, so that only if that late LTP synaptic changes happens, retrieval becomes easier. Okay, so any change occurring to this process will obstruct the retrieval process of memory, right? So now the early LTP changes which occurs as a result of strong frequent stimulation, as I have mentioned, occurs in the hippocampus, in the pyramidal cells of hippocampus with the help of Schaffer collateral. Okay, so now this is a Schaffer collateral. You can see glutamate will be released as the neurotransmitter there. Now the glutamate, for glutamate, there are two receptors present on the postsynaptic membrane. You have AMPA receptor and NMDA receptor. Okay, AMPA receptor, glutamate binds to both AMPA receptor and NMDA receptor. NMDA receptor is a calcium channel, and only if calcium enters through the NMDA receptor into the postsynaptic neuron, further changes occur in that postsynaptic neuron, which results in increased neurotransmitter release and further changes in the postsynaptic neuron. Okay, but there is a problem because the NMDA receptor is blocked by a magnesium block. Okay, so unless this magnesium block is removed, calcium cannot enter through the NMDA receptor. So what happens is that this is overcome by the presence of AMPA receptor. Glutamate, as I said, glutamate binds to both AMPA receptor and NMDA receptor. When glutamate binds to AMPA receptor, there is influx of sodium and potassium. So a little bit of sodium and potassium enters the postsynaptic neurons. That will produce a partial depolarization of the postsynaptic membrane. This partial depolarization but will remove the magnesium block. So when the magnesium block is removed, calcium will now able to enter the postsynaptic neuron. So our, what do you call it, a second messenger, you can call calcium as a second messenger, which ignites a lot of cytoplasmic reactions inside the postsynaptic neuron, enters the postsynaptic neuron. Okay, this is what happens in the early part of LTP. So I'll repeat once more. When upon a strong stimulation, frequent stimulation, glutamate will be released. Glutamate binds to both AMPA and NMDA. Okay, but NMDA is a calcium channel receptor, but the entry of calcium is blocked by magnesium. This block is removed by partial depolarization of the postsynaptic membrane with the help of AMPA receptor, which allows potassium and sodium influx when glutamate binds to it. So this is the first part of story. Now let us see what happens when calcium enters the postsynaptic neuron. Okay. So when calcium enters the postsynaptic neuron, it activates certain protein kinases. Calcium calmodulin kinase will be activated. Protein kinase C will be activated and phosphatases will be activated. You can see another same picture. Glutamate will be released. Glutamate binds to both AMPA and NMDA. As a result, the magnesium block is removed and calcium enters the postsynaptic neuron. This calcium activates protein calmodulin kinase. It also activates tyrosine kinase, okay, help of calcium. Now, once these protein kinases are activated, what does the protein kinase do? The protein kinase will phosphorylate the excess simply it will upregulate the number of postsynaptic receptors on the postsynaptic neurons will get increased. More synthesis of new receptors happens, and insertion of these receptors on the postsynaptic neurons will increase. Okay, and not only that, it will generate a retrograde signal generator, which is probably hypothesized as nitric oxide. This nitric oxide travels to the presynaptic neuron and causes enhanced neurotransmitter release. Okay, so now upon strong stimulation, there is increase of calcium in the postsynaptic neuron. This calcium activates a lot of protein kinases, and these protein kinases thus increase the number of receptors on the postsynaptic neurons, and also it increases neurotransmitter release from the presynaptic terminal to the postsynaptic neuron. In short, what happens is that activity in that synapses increase. Okay, so this is early, that retrograde messenger is nitric oxide, hypothesized as nitric oxide. Okay, this events to occur takes minutes to hours, and these are the changes occurring as part of early LTP. Okay, but what has happened is that there is increased neurotransmitter release, or there is upregulation of receptors for in that Schaffer collateral, with the help of Schaffer collateral. Okay, now further stimulation will result in consolidation of memory. That is what we have always heard about. Consolidation of memory or formation of memory traces, or called as something called as engrams, are formed in our brain. Okay, so this is what is the synaptic event occurring as part of late LTP, which will take some hours or sometimes some days or weeks. Okay, so now calcium, along with certain protein kinases which it has activated, it also activates adenyl cyclase. So this is something extra action done by the calcium. Along with protein kinases, it also activates adenyl cyclase, which increases the generation of cyclic AMP and protein kinase A. Initially, it was calcium calmodulin kinase, protein kinase C, and tyrosine kinase. Now, what the calcium does is it activates protein kinase A with the help of cyclic AMP. Now, what does protein kinase A do? It will activate MAP kinase pathway and generates transcription factors. To put it very simply, it generates certain transcription factors called as CMP response element binding proteins. Okay. Now, this transcription factors helps in increased protein synthesis. To put it very simply, it results in increased plasticity related synthesis of proteins, protein synthesis. Okay, which are the new protein synthesized as part of activation of cyclic AMP and pKa? These are more AMPA receptors will be synthesized, postsynaptic membrane, other proteins will be synthesized, which helps in synaptic transmission. Structural proteins will be synthesized. BDNF, that is brain-derived neurotropic factors, will be synthesized. Like that, more protein synthesis will be synthesized. That will help in growth of new dendritic spines, enlargement of existing spines, and formation of new synapses. Now, here you can see this is the whole story. This is the Schaffer collateral, which increases calcium in the postsynaptic neuron. This calcium initially activates protein kinase C and calcium calmodulin that causes upregulation of AMPA receptors and increased release of neurotransmitter from the presynaptic terminal, that is early LTP. Not only that, this calcium activates adenyl cyclase, cyclic AMP, and transcription factor. As a result, a lot of new proteins are synthesized, which helps in genesis of new dendritic spines and insertion of new receptors over there, and new collaterals will be formed, or new synapses will be formed. This is what is called as memory traces or engrams, or what happens as a part of consolidation of memory. Okay, so as you can see, memory formation is not a simple process. It involves lot of protein synthesis, lot of cellular mechanism. Calcium is needed, ATP is needed, proteins, lot of protein chemical factors are needed. Okay, so any problem in any of this process will help in destroying our memory. Okay. So now this is what is the basis of long-term memory, which is needed for our new synapse formation or new learning to occur. Okay. And this is continuously being formed in our brain or as a part of synaptic plasticity. As I have said, in the formation of memory processing, the first stage is encoding. How better we encode that information, that better will be the later on steps which is happening in our brain. All these steps which I described will be happening in our brain, and retrieval will become better. Now there comes the task of a teacher. Which I said, the students which are coming to us are mostly in the late adolescent age group, where the pruning is happening in our brain. Okay, so what our responsibility is there to give them the right kind of information, to provide them the right kind of stimulation, so that the pruning occurs in the most efficient and most appropriate and most adequate manner, so that students will be exposed to lot of environmental factors which can destroy their pruning process and can destroy the memory consolidation process also. Okay. So now a word about why we forget. Now, with all this great difficulty, we have created an engram and memory traces. But of course, there are instances where with great difficulty, we have recorded something in our brain, but after some time is lapsed, we forget about those factors. Okay, what is happening there is, as we have discussed before, degeneration of these memory traces if not continuously stimulated, which occurs as a process of long-term depression. Okay. Weakening of synapses with low frequency stimulation. There will be lower calcium influx and protein phosphatases, certain other protein phosphatases will be activated. That will cause dephosphorylation or downregulation of receptors and internalization and decrease in the number of receptors in the postsynaptic memory. Now, this process is also important along with long-term plasticity. Okay. Why it is important? Because it helps us to clear out the irrelevant memories or irrelevant memory traces which has been created, which helps us to clear out and refine the neural circuits, which is a part of evolution or development, helps us to create new traces or new circuits and enabling new learning and prevention of saturation of long-term potentiation. Mainly occurs in all those areas which are a part of this learning and memory process. So along with long-term potentiation and long-term depression is also an important process. So forgetting is not unnatural. It is very natural process, and it is also required so that our brain becomes more refined and more clear. Okay, and only those things which should be maintained will remain in our brain. Okay, right? So understanding this, if you understand this physiology of learning, of what all we have discussed, understand the process of synaptogenesis, synaptic pruning, long-term memory processing. Okay, and what is the importance of pruning and what is the importance of discussing this in viewpoint of the students which we are catering to is very important. I hope after this session is over, you will view you will approach your students in a different angle. Okay, right? So now, what is the difference between learning and memory? There is a subtle difference. Learning is acquisition of new experiences, neural mechanism by which individual changes his behavior on the basis of past experiences, what is called as learning. What is memory? Memory is storage, retrieval of this information. Okay, without learning, memory is not possible. But without memory, learning can be done. Only thing is that it cannot be retrieved. So not all learning is transformed into memory, because encoding, it depends upon proper encoding, proper consolidation, and proper storage. Then only a learning process will be converted into memory. Our students always also complain that I studied, madam, I studied very nicely for the exam, but I could not remember anything. Okay, there is a definite answer which you can give to the students. Why? Because your memory is not converted into long-term memory. See, short-term memory or early LTP will be easily destroyed in your brain. So only you have to adopt with certain measures so that your encoding is proper. Repetitions is what is important. Revisions are important for converting our short-term memory into long-term memory. Okay. So there is no memory without learning, but there is learning without memory. That is the connection between learning and memory. Okay. To ensure memory, learning is important, but it does not ensure that you have learned. Every learning process will be converted into memory. You have to put some effort, do repeated learning, then only learning process will be converted into memory. And only if memory it is stored or consolidated in the brain, can you retrieve it back when you need it. Okay. So that is the connection between learning, memory, consolidation, synaptic pruning, etc. Right? Now let us come back to our role as a teacher. Now we have understood what happens in our students' brain at the time of learning process or memory process, and their age group, what is the problem with that age group. In the physiology point of view, somewhat we have understood. Okay. Now, as a teacher, what we are doing? We are teaching our students a lot of things, right? Now, expecting a change in behavior happening to the students. Okay, we are teaching. Teaching is a process by which an expected change is expected in the student's behavior. Now, we have to function more as a role of a facilitator than a teacher, and be more an effective teacher than an efficient teacher. Anybody who is in medical education will probably understand the difference between efficient teacher and effective teacher. It is not that we complete the portion allotted to us. It is ensuring that what we have taught has really produced a change in the student's knowledge, student's skill, or student's attitude. Okay, so that is what is prescribed in our competency-based medical education. We have to be effective teacher rather than be an efficient teacher. Of course, we always have that burden of finishing the portions in front of us before the course is over. That has to be ensured. Along with that, let us just ensure along with that, we also take few steps to ensure that whatever we are taking in the class is produced a change in students' behavior or their skill or attitude. Okay. And that can happen if the encoding process occurs accurately or properly in the student's brain. And for encoding, motivation, key factor is motivation. Unless the student is not motivated enough, that is probably what we are all facing. Till the time they clear the entrance exam, they are all well motivated. So I always envy the students who are the teachers who are teaching in this coaching classes. They get the high motivated students. Okay, more than the teachers, the students are motivated to learn. Okay, but the students, unfortunately, coming to us, all the motivation gets wiped off somewhere in the lane, and the motivation factor is somewhat reduced in our students. So how can we increase the motivation level in the students? Unless that is done, the encoding process will not be proper, and the stages of memory processing will not be accurate. Okay. So the main role of the teacher is in planning and creating a condition which is favorable for learning, and motivate our students. Okay, but nothing can replace the knowledge of the teacher and the self-effort of the learner. Unless the learner is motivated, however knowledgeable the teacher is, will fail to impart the knowledge to the students. Okay. Now there is a publication coming from Harden and Crosby, the famous educationalists. in the newer concept, they have described the roles of a teacher. There are 12 defined role of a teacher. It is not only imparting knowledge, it is not only information provider, but the teacher, all of us are actually taking part in all this role without our knowledge. In our subconscious level, we are carrying out all these activities. Harden and Crosby has just put this in black and white. That's all. Okay. So the role of a teacher is not only as an information provider, but be a role model, facilitator. We are being an assessor. We are planning their curriculum. We are developing the resource. All this is a part of teacher's role. Okay. Now, coming to the teaching learning methods, which we are adopting for imparting this learning process or memory process to occur, there are various types of teaching learning methods available. Okay. So, first, there are certain concepts I'll discuss before we move on to the proper teaching learning method. No single method is considered as best. We have to adopt the suitable one for that suitable audience, for that suitable objective or competency, for that particular situation, depending upon our resources. So no single method is considered as best. It should be aligned to our competency and objective, suitable for the learner. It should be motivating and feasible for us to implement. And it should suit the teacher's personality and assess. If something is not suitable, you are not comfortable, better give it to some other person who is comfortable in handling that teaching learning methods. I'll be discussing few teaching learning methods here, which will make our learners more motivated, or will help us to bring that boredom of long hours lecture classes. As you know, there are various types of teaching learning methods. Teacher assisted methods are there, teacher non-assisted methods are there, or methods for all your domains, cognitive domain, psychomotor domain, affective domain are there, both in teacher assisted and non-teacher assisted. Okay. Now this will be covered in your basic course classes. I'm not going into detail about this. You have got, based on the group, there are large group methods and small group methods and individual methods suitable for single individual learning. Okay. So all this we are aware. Now, what is not routinely practiced in our traditional curriculum, what is being given importance in CBME-based curriculum to make our classes more interactive and more motivate the learners is what I'll be discussing. Just one slide on each of these teaching learning methods. Now, why should we do that? Why should we see? We were happy with the curriculum going on, everything was smooth and fine. Now, why should we try to make our classes something different? Okay, so there is a graph which is showing, which is shown here, the lecture without any student activity in between, and a lecture with a student activity planned in between. Okay, so we all know that there is an attention span for every person who listens to a lecture. How much is the attention span? Previously, it was said that 45 minutes. No classes should be beyond 45 minutes. That was mentioned previously. Later on, it came down to 20 minutes. After every 20 minutes, there should be something to break that monotony, to motivate the student and then continue. So 20 minutes was mentioned later. But now if you see in the newer generation where we are in the surrounded by so many in our social media, you have reels and shorts. Nothing is going more than 1 minute, isn't it? So the attention span has progressively come down. We also do not like to hear to a lecture more than say 20 minutes or 30 minutes. Okay, unless that our attention is our monotony is broken down and it is recollected. Okay, so to maintain that attention span throughout your lecture duration, some type of interaction or student activity has to be planned. Okay. So that is why we are discussing this newer teaching learning methods, and CBME also prescribes us to move towards a learner-oriented teaching process than sticking on to a teacher-oriented learning process. We have been asked to be more student oriented, active learning, problem solving methods have to be implemented. Okay, and more skill teaching has to be incorporated. All that is advised in part of CBME. So these are certain innovative teaching learning methods which I'll be just discussing. Just one slides on. So many are discussed there, but I'll just enlighten you what it is and where you can use it. Okay, a big list is shown in the slide right now. Brainstorming, bus group, snowball, fishball, like that. Now, certain are suitable for large group. When you have a large group lecture, in between you want to plan an activity, some of these can be utilized. Okay, as we discuss, you will understand. There are few which is suitable for cognitive domain or your lecture classes, few suitable for your psychomotor domain, and few suitable for affective domain teaching learning methods. Okay, so we'll start one by one. First is brainstorming. Everybody knows brainstorming. So suppose you are taking a class. Now I am taking a class. I started from synaptogenesis, later on moved on to synaptic pruning, and now then moved on to synaptic plasticity. Then I discussed regarding long-term potentiation and long-term depression. Then I discussed regarding the difference between learning and memory. Now I am discussing the difference between teaching and learning and various innovative methods. Now what I have done, I have just broken that monotony of lecture. Took just 2 or 3 minutes to go through whatever has been discussed so far. Okay, so now this is one way of breaking that monotony. Another way of breaking the monotony is brainstorm. Brainstorm means you put a question to the large group and ask them to come out with some answers. Okay, it is brainstorm. Like for example, now this is best suitable for something like that. A general thing. We'll just a general thing. Suppose you have a dog menace in your campus. You have a lot of dogs around in your campus. Okay, you put a question to the large group of students and ask them to bring out ideas, generate ideas. How can you control dogs in the campus? Now, why I took that specific example is that now in brainstorming, there is no pre-preparation and you don't have any defined solution. You need various types of solution from a large group. Okay, out of which we can select some and implement which are the feasible ones. So brainstorming is utilized for that type of teaching. Okay, so but it has to be planned just 5 minutes in between your lecture. Some you can ask them brainstorm. A concept can be put to them and ask them to brainstorm on that concept and come out with a solution. If you come to your clinical medicine, okay, a 20-year-old female came with pallor, right? 20-year-old female came with pallor. Now you put that question to the group, right, and ask them to come out with their diagnosis. So that is, so one will say anemia, another will say some other answer. Okay, so that is brainstorm. So, but that has to be time limited, and the teacher should have control over, because once you leave that large group with a brainstorm, a lot of buzz will happen in the class, and it has to be started with control and has to be controlled through the process. And in the end, all those points which are generated should be noted down and it should be listed. Okay, so that is brainstorming. Next is snowballing. Snowballing, it is little bit of a tedious task. Now that can be little bit more complicated than brainstorming. Brainstorming will be easier for us to implement in a large class. Snowballing, what happens is that suppose you have few students, suppose 30 students are there, or 30 or 40 students are there. Okay, you put one idea to one student, you put one question to one student. Okay, give question to all the students. So they will think about an answer to the 20-year-old female came with the pallor. So everybody will think separately first. Then you ask 2 of them to pair, think and pair, 2 of them to pair. So this is what is shown here. First, a solo brainstorming happens. Then 2 of them will pair together and a pair discussion will happen. So both of them will come out with their answer. Okay. And finally, what happens is that both will discuss and they will say, yours will have this problem, yours will have this problem. They will come with a single answer. Now these two will form a join with another pair. Okay, and similarly, a snowball, just like a snowball is gaining its momentum and gaining its size, the student small group will also gain its size. Discussion will continue from small group to large group, and whole class will be taken into account. And finally, one answer should come from the class. This is what is called as snowballing. Not an easier method to implement in our setup. What we can do is think, pair, and share. That can be easily implemented in a large group class. You can give a question to the students, ask them to think separately, then pair, discuss the answer, and share. Okay, so that is think, pair, share. One step in the snowballing process. So this is brainstorming and snowballing. Then there is something called as bus groups. Bus group is also similar, forming a small groups in a large group. Okay, and they are discussing that particular point. It is similar to think, pair, share. Not snowballing is not happening there, but a small groups will discuss among themselves and come out with the answer. That is bus groups. Okay, this all you can plan a 5-minute activity in your lecture class. So this has to be incorporated in your lesson plan before you go out for your lecture. Okay, so that is bus groups. Then something called as fishbowl. Fishbowl is again an activity where you have a large group of students. Learner center, instead of you taking class, you can put the question to a group of students forming an inner circle. Those students will discuss that particular concept or that particular topic, understand that, and learn that. You have an outer circle who will be watching the activity which is going on to the inner circle. Just like you have a fish in a bowl, the fish will be roaming around, and you, spectator, audience, will be watching the movement of the fish inside. Here, the activity occurs between the students inside the circle, and there will be a group of students who will be just watching outside. But they also learn from this activity. They are not actively participating, but they are passive listeners. They will be passively watching, but learning occurs. Instead of you taking this class, the inner circle students takes the class or do the learning activity, which will be observed by a group of students sitting outside. Later on, you can reverse the role. Okay, so that is called as fishbowl technique. So far what we have discussed is that brainstorming, snowballing, then bus groups, and fishbowl. The first three can be very well or think, pair, share. First these can be implemented in a large group in a lecture class. Fishbowl can be implemented in a more small group discussion class. Okay. Now, next comes three important concepts, that is team-based learning, case-based learning, and problem-based learning. Before I show you the slide, I'll put it in a very simple term. What is most often these three teaching learning methods are discussed as the same one, but they are not the same. They are different. Team-based learning is different. Case-based learning is different. Problem-based learning is different. Okay. Now, what is the basic difference between the three? I'll tell you. We are more familiar with case-based learning. What is case-based learning? We often use this technique for our teaching learning process. For example, if you are teaching hypothyroidism, then you move on to discussing using a case, you discuss that particular topic. That is case-based learning, which will be completed in one session. Just teaching hypothyroidism, you are using a case and discussing the hypothyroidism. And this problem-based learning, team-based learning, I'll tell you at the end. How different is problem-based learning? Problem-based learning does not get completed in one session. It needs multiple sessions. Okay. Instead of giving the topic as hypothyroidism, you first what you give is you give part of the clinical, the day one when they come to you, when the student comes to you, okay, you give part of the clinical history. And we ask the student to go back and refer and come out with various differential diagnoses. So second session, they will be invited and they will have 2 or 3 differential diagnosis in their hand for the part of history which you are given. Now, in the second session, what you give is that you complete the history and give certain general investigations which are done as part of this case. So again, from 3 or 4 differential diagnoses, now they will come down to 1 or 2 differential diagnoses. That will be session 2. Now, again, in session 3, what you do is that you give the definitive clinical diagnosis for that particular case. So from the day 1 where they were having 5 differential diagnoses, day 2 it has reduced to 2 or 3, and in day 1 it has been reduced to 1 diagnosis. So from a problem they have come to a diagnosis. Okay, so that is problem-based learning. So it will take multiple sessions and lot of time and effort and research has to be come from the learner's side. So it is not an easy teaching learning method to implement. What is more easy for us is case-based learning. Okay, similar is team-based learning. In team-based learning, what happens is that again, a pre-class preparation. There should be a pre-class preparation. This is a short diagram showing the process of team-based learning. First, there will be a pre-class preparation. Material resources will be given by the teacher. So learning does not happen in the class actually. Before the class, the student has to prepare and come. And when they come to the class, first an individual test will be given to each of the students. So first the student in the class appears for a test. So that is called as IRAT or Individual Readiness Assurance Test. Okay. Now, next what is that? Based on the Individual Readiness Assurance Test, the students will be made into small teams. Now, they'll take the same test. Suppose it's a 10 MCQ. Individually, they have made 5 MCQ correct, 5 wrong. Now, they are made into teams of 3 or 4. Okay. Same test is again given to the students. So now, because of their group activity, what happens? From 10 MCQ, instead of making 5, 8 or 9 will be correct because there is a group activity. Okay, so that is team readiness assurance test. And then what happens? Then the class, then what is done is that clarification of the doubts which the students are having will be done in the class. This is something in line with something called as flip the classroom, because before the class, the learning has occurred. In the class, what is happening is that only discussion is going on and immediate feedback and clarification to the students is given in the class. Okay. And later on, the student will be given some task or some problem solving activities so that their concepts will become more clearer. Okay. And the session will be closed. So theme-based learning is also not similar to case-based learning, not similar to problem-based learning. It is forming a team and interactive for doing a collaborating learning and your higher level of cognitive domain. Just recall is not sensitized here. Higher level of cognitive domain like analysis, problem solving, etc. will be motivated. Okay, so more of student-centered activities involved in problem solving and also in team-based learning. Case-based learning, just a motivation from just to attract the attention of the student. A case is brought and they will be so that they will be more interested in the class. That is, so this is the basic difference between case-based learning, problem-based learning and team-based learning. Okay. So we have discussed the differences. Problem-based learning again occurs, there are a lot of process or homework to be done by the teacher. It has to be conducted in 2 or 3 sessions at least. And only at the end of the session, the student will get the whole concept of the topic. Okay. So there are differences. It's available everywhere. The differences between problem-based learning, case-based learning and team-based learning. Okay. Now, again, another concept which is coming up is peer-assisted learning, because we are always catering to a large group where the student strength is more than 100. It is not possible for teacher to give individual attention to the students. Okay. So what is now promoted is something called as peer-assisted learning, where a group of peers or students who are little bit high scorers can be recruited and they can be used for the teaching purpose out of the academic hours, or during you can schedule a timetable, you can schedule a time for in your normal timetable also. Use the students itself for teaching low scorers. That is peer assisted learning, also coming up in a big way. Okay. So these are various innovative teaching learning methods which are not there in traditional curriculum, but now being promoted in CBMP. Okay. Now, in the recently we had a CME conducted by a Department of Anatomy in our Government Medical College, Kottayam, where a nationwide competition was conducted for concept mapping for undergraduate and postgraduate students. Concept mapping or visual mapping. Okay. Now, this is can be promoted in our students. For, as you know, there are different types of learners. Auditory learners are there, then visual learners are there. Okay, so concept mapping can also be adopted. When you teach a difficult concept, what you can do is you can plan an activity. Okay, so you have got your museum project or library project to be done as part of CBME. What you can do is that, suppose your topic is memory, ask them to prepare a concept map of memory. The main concepts of memory has to be connected together. Okay, connected together, and a map has to be created. Whenever you have a difficult concept taught to the students, after the class, you can ask the student to submit a concept map of that particular topic as an assignment. It will be just better than, so different concept map will be prepared by the students. So it will be better than just writing it down as question and answer. A concept map, the whole concept can be concised in one single sheet of paper. So this is what is called as concept map or mind map or conceptual diagrams. Different names are known. It is visual mapping of the basic concepts. Okay, in bits and pieces connected together. Okay. Another innovative teaching learning method, which is right now not actually implemented, but it is in the part of various research, educational research programs, is game-based learning. Like games like puzzles, jigsaw grouping, then treasure hunt, etc. are being utilized for learning purpose. It is being done as a educational research program. Not actually implemented. I don't think it is actually implemented in any of the colleges as part of curriculum, but something you can think of in an innovative way which can generate interest in your learner. You can motivate point. What we have learned is that the most important factor in learning the method, the encoding memory trace will be created in the brain. Okay. Narrative writing is something which can be utilized for effective domain, for your effective domain. The patient story bedside, in the bedside clinics, when the student is writing down the case history, it can be in the form of story rather than in the form of your format. Format has to be there. But to generate interest, sometimes you can utilize this narrative story. The storytelling, the actual events in the chronological order from the patient mouth is written down in the student's notes as a story rather than in the form of format. That is narrative writing. And reflective writing is something which we are utilizing now as part of EdForm module and as part of developing that affective domain in the student. The student has to undergo a learning process, a scenario. Student has to write down in the following headings, right? What happened, so what next? Okay, what happened, so what happened? What was the learning activity which has happened? So what change it has produced in you? And so what next? What will be a change in behavior which will be produced by you in your future encounters with the patient? Okay, so that is reflective writing, a very effective method of teaching affective domain if done in a proper way. But what I have seen in our students, I hope it is universally applicable to all students, is that all this reflective writing, narrative writing, etc., concept, not everything is now not done as a part of student activity. It is done as a part of chat GPT or AI. Okay, so that is our task, next task. Okay, to disconnect this AI from our students. How can you do that? Okay, so that I think everyone has to think about it. If all is done in a classroom under the control of the teacher, a lot of learning process can be motivated. Okay. Next to concept is self-directed learning and self-learning packages. Both are looking similar. Self-directed learning and self-learning packages, both are looking similar, but both are not the same. In self-directed learning, what happens is that the out of the it is designed for something co-curricular activity. For example, it is more suitable for our most adopted by us teachers or our PG students. Suppose they want to learn take up a course of AI in medical technology or medical education. It is not their curriculum. It is just because of their interest they want to learn about AI. They get hold of what do you call it, the learning program. The objectives they will select, and everything is decided by the student. Okay, and what we give is that just the direction. So the need is by the student, the goals are formed by the student, then the identifying the materials is also done by the student. Okay, and everything is done by the student. Assessment will be available, is also taken by the student. Like our students are going for this PG NEET entrance exam, PG entrance exam. Okay, so they will go and find out, okay, now this will be better than the other resource material, DAMS will be better than Bhatia or something like that. They will design their time frame, they will select the resource material, and they will select the assessment exam. Now it is self-directed. Everything is done by the student. What is available, it will be accessed by the student and given by the student. But self-learning packages is somewhat different. That is what is more possible for us in our undergraduate. Okay, we can design a package which will be available. Suppose research methodology package is available. It is available as a program in our institution. For example, a Google Classroom you have, or a Moodle portal you have. You upload a package for research methodology for undergraduate, and you say that now this package has to be accessed during of this phase group. Phase 2 students all have to access this package. At the end of the phase 2, you will have an exam going. So the student at their comfortable time will access that package, study that, and give that exam. Okay, so there is a liberty given to the student, but everything is designed by the teacher and it is made available by the teacher. Only thing is that the teacher presence is not there, and only the assessment part is done by the teacher. So that is what is it is mandatory in self-learning package somewhat, but it's not mandatory in self-directed learning. That is basically the motivation is completely done by the student, whereas in the other it is somewhat done by the teacher. Okay. So far we were discussing the various innovative teaching learning methods for cognitive domain and for affective domain. Okay, now we'll move on to certain teaching learning methods which are not regularly used by us, but which can be used by us for imparting skill training or psychomotor domain. Okay, psychomotor domain. 1 minute preceptor. I hope everybody knows this is applicable for this intellectual skills as a part of bedside clinics. You have a bedside clinics class. At the end of the bedside clinic class, 1 minute should be utilized for getting a commitment from the student, for probing the students for giving the evidence for that commitment, teaching them certain general rules, reinforcing what the student has done right, and correcting the mistakes of the student. Or 1 minute, or in short, 1 minute should be devoted for summarizing whatever clinical discussion has been done before that. Okay, so that is called as 1 minute preceptor, used for imparting intellectual skills. Okay, for imparting psychomotor skills, you have DOMP. What we do is that whenever we teach a skill to the student, for example, examining the radial pulse, okay, we show the student how to examine the radial pulse. We demonstrate and we ask them to do, and we just leave the scene. Okay. Now, instead of that, what we have to do is that you demonstrate, you observe what the student is doing, you assess the student when the student goes wrong, correct the mistake, and finally, the student should be doing without your assistance. So this is DOAP. Okay, DOAP. Instead of only demonstrating, you have to be there, observe what they are doing, assist them when they are doing, and finally, they make them capable of doing it without your assistance. Okay, so this 4 steps has to happen when you are teaching psychomotor domain. Another step, which is a method, which is again prescribed for teaching psychomotor domain, is Patent's 4-step approach. Okay, Patent's 4-step approach. Here also similar thing. Only thing is that when the teacher is demonstrating, the teacher first demonstrate the steps of radial pulse. Next, the teacher spells out the various steps of examination of radial pulse. Okay. Now what we talk, the trainee through. Teacher spells out. Now the teacher does it when the trainee is asked to spell out the steps. Okay. And finally, what is done is that the trainee does it without assistance. Okay. So I'll repeat once more. This is another method of 4-step approach. First is the teacher demonstrate without any commentary. Next, the teacher does the steps and spells out each steps or gives the step-by-step commentary. Okay. Now, third thing what is done is that the trainee will spell out the commentary, spell out the steps, whereas the teacher does it. And final step is that the trainee will does the step without your assistant, without your spelling out the, or without your commentary, this trainee will does. Okay, so this is Patent's 4-step approach, this all is different methods of teaching psychomotor skills, but time consuming factor is always there. One-to-one teacher interaction has to be there. There are a lot of hindrances, but more effective and student-centered approaches. And last word is something, a new concept. It's not a new concept, but it's a concept which used now and then by some innovative teachers, but now accepted widely, is cinema education. Using film clips or clips for imparting effective domain or communication skills to the students. That is the only way where you can effectively teach communication, either by direct demonstration, role plays, or by cinema education. Okay, utilizing cinemas, clips, not full cinemas, but clips which are needed for us can be utilized for imparting attitudes and communication skills. Okay, to the students. So that is regarding innovative teaching learning methods. So in summary, what we have discussed today is that we have started from physiology and ended with medical education. Okay, started with synaptogenesis. At the time of birth, we are born rich with maximum number of neurons. Later on, maximum number of synapses happen. Later on, depending upon our activity and influences, the number of neurons, synapses comes down, and those remains which are actively utilized remains, and not utilized just vanish off from our brain. That helps us in learning and memory. And memory is a very active process in our brain, which involves lot of protein synthesis, lot of energy consumption, and it occurs in various steps. Any problem, any of the steps will help hinder our memory processing. And we have to understand that the students which are coming to us are in the evolving process, and to motivate them is our task. Unless they are motivated, their learning becomes improper. To motivate them, you have to think about this innovative teaching learning methods, which are not there in the traditional method, which are now being promoted in CBME. But we have to be careful that it should be suitable to the teacher, suitable to the learner, teacher's personality, our resources, our learning environment. Everything has to be taken care of before we adopt to any of this innovative teaching learning. Okay. Thank you, madam. So that was what I had to discuss. If there are any questions, I am happy to take up. Yes.
So this presentation is open for the open discussion. If anybody has got any doubts, you can clarify. With the Dr. SARITHA.
Hello, Sharitha. Hello.
Madam.
As
Madam.
very.
Good evening, madam.
Can you see me? Can you see me? Can see me, no? Can you see me?
Yes, madam. Madam, I can see you.
That is a very lucid, very nice presentation. As all yours are actually. And it's a very difficult thing you have taken us through. But one problem that we teachers really are facing now. I mean, we cannot be solved by all these things. All these methods are good. But the thing is, the time available with the students. All this requires some amount of time with the students. So yes, with the 8 months, we are finding it rather difficult to catch up with all this. Anyhow, the small group discussion, all this, some of this like cinematograph are being applied now is very useful. But in spite of all this, we find that most of the time the students are present day. They are looking for what is easier. So very often they come out with what is that from the YouTube or some online material. They prefer that than writing down our notes or listening to us and reproducing, because that is easier for them. If you ask them to do a seminar, they just download some PowerPoint and present that. So any solution for this thing, that is to motivate them more. Since you are in the teachers training program also, I'm asking.
Madam, we are also finding it very difficult.
We are telling them, but
so when we discuss it along with a group of teachers, which we interact, different groups, what feasible suggestion which has come up is that rather than giving them a homework, that when you give time for them to go back and do the work and come to the class, what they do is that they happily give that work to the ChatGPT and they bring it to the class. Okay. So rather than doing
you mean assignments, is it?
Assignments or seminar topic. So it is better to do it in live class itself. That is the only way. You give them some time in the class, short topic. That is applicable only for short topic. Some time you give it in the classroom. Ask them to prepare in the classroom. At that time, they can refer textbook. And whatever they have prepared in the short time, they can present it in that class itself. Suppose you have 2 hour free batch time. Okay, so at least half an hour you can give it to them for preparation in the class. And next 1.5 hours you can ask them to present. That is the only way, madam. If you ask them to come and prepare, it is what we have also found is that this just does not serve the purpose.
Seminar they are doing well. Seminar they are doing well. We have one seminar for each student that they are doing rather well. Most of them, majority of them, and assignments also they are doing well. Like you said, when you give them work, that is better. Okay.
So then what happens is that the number of activity will get reduced because all these are time-consuming process. Yes. Everything cannot be done under the presence of teacher. So the activity number comes down. Okay, but whatever we are doing will be effective.
That's correct. That's it. And thanks to Believers Church for making it online so that all of us could attend. So Annamma, special thanks to you and to your principal. Okay.
Thank you, SARITHA, for your excellent but vast presentation. Actually, the normal, that is, the brain is an incredible organ, has got an ability to reorganize itself, forming new neuronal connection, you already said, and that is important for enhancing learning. And not only learning and memory, it also involves, this neuronal connection is important. It develops after the injury to brain also. Brain concussion, etc. After that, the brain regains its activity because of this neuronal plasticity. And that is then you explain about the synaptic plasticity and the importance of LTP. that is, and also I want to tell you that this LTD, what is the LTD? That means a long-term depression. That is actually a bad plasticity, isn't it? So that is synaptic plasticity, then structural neurogenesis. Regarding neurogenesis, the number of neurons are actually stable, but that is, it is said that the hippocampus has got an ability, I don't know.
To regenerate. Yes, ma'am.
Hippocampus has got an ability to produce more and more neurons and connections. And it is said that after age of 60 years, that 1/3 of our neurons in the hippocampus are new one. That is one that is in book, it was documented. Then the neuroplasticity is actually seen across our lifespan. That is starting from adolescent and all, and it is saying throughout. It is permanent throughout. It is going on or happening throughout our life, and then but it is modified by certain stimuli in the form of learning, repeated learning, and also other factors you already mentioned. Then regarding the best teaching learning method, active learning, everything well explained. And also you that is said about how to improve cognitive, affective, and also psychomotor domains. And that because so much of them are practicing in our college. Then Saritha, could you able to tell me that is some advice? So because neuronal plasticity is the basis of the learning of the students and memory of the students. Can you say some factors which can that is increases or decreases, especially hormonal factors or exercise or role of antioxidants and food habits, which are
Hello.
so memory and learning are going in that already you explained. Then so it is the daily habit they have to follow is, one is the repeated learning, and also they have to, so I mean, if they got time, that is a time spent for solving puzzles. And exercise is very important because that is bone derived neurotropic factors are more produced during that is exercise, and also during sleep, and also that is when you undergo sun bath. So healthy food is very important, and also sleep, and you are already said, and also learning new skills, and also students has to socialize and stay organized and set goals. That's important for that's an important advice for the students to improve their memory and learning. Thank you, Saritha, for an excellent presentation, and that is in the name of we are very happy to be with you. Thank you.
Thank you, madam. Thank you so much.
സരിത വളരെ നല്ലതായിരുന്നു. ഞാൻ അതുകൊണ്ടായിരിക്കും ലോകം മുഴുവൻ പറ്റുന്നിടത്തൊക്കെ ഇട്ടു നമ്മുടെ ഫിസിയോളജി ഗ്രൂപ്പില്. പക്ഷെ കുറച്ചു പേർക്ക് കയറാൻ ഈ സമയം ശരിയാവത്തില്ല. അതുകൊണ്ടാണ്. ആൻഡ് ഐ എം താങ്ക്ഫുൾ ദാറ്റ് ജമീല മാഡം ഈസ് ഹിയർ, യു നോ, സീനിയർ പേഴ്സൺ ഫിസിയോളജി പ്രൊഫസർ. ജമീല മാഡം ഉണ്ട്. ജമീല മാഡത്തിന് യു ക്യാൻ ടോക്ക്, സേ എ ഫ്യൂ വേർഡ്സ്. ആലിസ് മാഡം ഉണ്ടായിരുന്നു, ആലിസ് പോയി. ആൻഡ് സരസ്വതി ഹാസ് കം. പ്രദീപ് ഈസ് ദർ, പ്രദീപ് തോമസ്. So they, in spite of no range and all, they had joined actually. So Pradeep and Jamila madam, can you say, speak?
So by which finish?
Am I audible?
Yes.
Am
Yes.
I audible?
Yes, madam.
Yes, madam.
It was a wonderful talk, especially regarding the plasticity. I was always worried about what will happen. See, we studied that there is no growth of these neurons if an injury happens like that. But now it has been proved that isn't it? It can formulate new synapses and so much boon to the patients also, isn't it? So what is your opinion regarding that? If something happens, some injury of the neurons happens. in the neurological case, for example?
Madam, hello.
Hello?
Madam. Hello.
What? Once an injury happens, how this plasticity and this help in the regaining capacity of the patient?
Madam,
regarding
it is documented that in certain part of hippocampus, as VIJAYAMMA madam has said, certain part of the hippocampus, the neurons has got a potential to regenerate. So new neurons can be created from those regions.
Yes. That's a very helping information, isn't it, to the patient?
Yes, madam. And the neuronal recovery definitely is based on a lot of other factors also, madam, like the growth factors, neuronal growth factors, neurotropins, BDNF, etc. helps in neuronal growth.
Yes.
So this is
And
what I
we
have
will
to share. This is my limited knowledge, madam.
It was a very good talk, dear. Anyway, for all the organizers also, I congratulate all of you, Dr. Annamma, your principal. Thank you, dear. Thank you, dear, so much. Thank you.
Actually, this Zoom is arranged by it is a plan of our Dr. George Chandi sir, and also great pain is taken by Dr. Jijo. He is present here, the pediatrics professor, and also Riju sir, and some other persons, many persons, technicians and all clinicians. So we owe thanks to them also. And it was a thank to all the participants, especially from different places. So then all physiology and anatomy, some of the anatomy persons also have attended. Thanks to all who participated. Key persons, Jijo sir.
Ma'am, it was an excellent session. Very innovative ideas have been passed on, and inquisitive thoughts has to be there, wherein how we can also train ourselves in the coming future. And I'm sure all the teachers who have listened to it will try to impart it in their teaching methods also. Thank you so much for joining us today, ma'am.
Thank you, sir.
Madam, I have one suggestion. That is, the neoneurogenesis occurs in hippocampus and olfactory mucous membrane. So I think we may have to do research what triggers these neoneurogenesis in hippocampus and olfactory mucous membrane, even though it is not possible in other places. We may have to find out the factors favoring that, and that factors may be researched whether they can be put to use without harm in regeneration of spinal cord neurons, etc., following injury.
Thank you, Dr. Pradeep. Thank you for all attending this program.
If there is no question, shall we wind up the session?
Yes, sir.
Okay, thank you all.